Okay, so then we will move on to our next presentation by Sonja Stojanovsky. And Sonja is going to talk about local network architecture in children with mild TBI. Sonja is a PhD candidate at the University of Toronto in the Department of Physiology, who studies the effects of mild traumatic brain injury in childhood on white matter microstructure and organization, and the role that white matter perturbations play in attention problems, which are common sequelae of TBI. So Sonja, go ahead and start, please.
Thank you so much for that introduction. Can you all see my slides? I'm using presenter view for the first time, so okay. All right, so I'm going to get started. So my talk, as Robin has already introduced, is about structural networks and mild traumatic brain injury in youth. And as we've heard many times today, more than half of all traumatic brain injuries occur in youth below the age of 24. However, the plasticity of the young and developing brain does not confer protection. And up to 29% of children with traumatic brain injury have reported impaired executive functioning up to 2 years post-MTBI. However, as we've heard many times today, it's very difficult to predict which youth go on to have persistent impairment. So we are particularly interested in attention problems and MTBI. Attention problems in particular are thought to be a common sequelae of traumatic brain injury. And a recent systematic scoping review performed by myself and my colleagues found that the prevalence of new attention problems and ADHD diagnoses after traumatic brain injury ranges between 5 and 53%, so quite wide, but on the higher end of things is something to be concerned about. We're also interested in attention because attention supports higher cognition, including thinking and problem solving and attention problems, which can negatively affect their social interactions and their ability to perform well at school and in the home. So for all of these reasons, we're interested in studying attention problems. So diffuse damage is caused by the rapid acceleration and deceleration that happens during the brain injury. This causes torsion within the brain tissue and primarily affects axons. And the way that this can affect axons is quite varied. So in the figure on the left here, we can see that some of the ways that axons can be affected is by demyelination or by traumatic axonal injury. You can also have excessive myelination, which is where the myelin that was on a axon that has been injured by the brain injury kind of sloughs onto another myelinated axon. And so for these reasons, we're very interested in studying the white matter. So in this study, we will attempt to measure disturbances to the white matter with something called fractional anisotropy. So fractional anisotropy comes from a tensor model of diffusion imaging. It essentially measures how freely water is able to move in one direction. The more ordered the directionality of water movement, the higher the metric. So on the left-hand side here, we see something that would produce very low FA, and this would occur in the CSF or in areas of the brain that don't have large white matter tracts, whereas on the right-hand side, this is very directional water movement. We would see something like this in the corpus callosum. And this metric is interpreted to quantify axonal integrity, though that's a bit contentious. But this is the metric that we're going to be using. So there are many different types of white matter, and we believe that the length and location of white matter contributes to its vulnerability to traumatic brain injury, and I'll explain why in a minute. So there are many different types of white matter. There is deep white matter, and there's also superficial white matter. So deep white matter includes fibers that we've heard a lot about today, such as the corpus callosum, which is a major commissural fiber. However, there's also this other class of white matter that is less commonly studied, and this includes U fibers and short association fibers. And these fibers are really important in the connection of the brain within the hemisphere, and they specifically connect from gyri to gyri, which is where they get that U-shape pattern from. And so the reason that we're particularly interested in these short association fibers or superficial white matter type fibers is because previous studies have found that injured axons are often observed in an area where tissue density changes. And this makes sense because if densities, they're going to move at different rates and cause damage to the axons that pass through them. We also know that this type of white matter has very protracted development and is continuing its myelination even into the 3rd decade of life. So it's one of the final white matter areas to complete its development or to complete its myelination. Also importantly, these short association fibers subserve local connectivity. So I mentioned that they are very short and they're U-shaped and they're connecting basically the gyri of our brain together. So they're very important for the connection of regions of the brain that are close together. And they specifically serve this purpose, I'm sure in addition to others. Finally, our previous research established the susceptibility of these fibers to traumatic brain injury and also their importance in attention and processing speed. Finally, the development of local community structure, which we believe these fibers are very important in, is crucial to executive capability. This study by Baum in the Philadelphia neurodevelopmental cohort found that the modular segregation of the brain, of the structural connectome, is what mediated executive function development in youth. So our question is how MTBI perturbs the local components of the structural white matter network and how these disruptions confer to the risk of attentional impairment. The way that we're going to look at this question is by looking at local connectivity in 2 ways. The first is by the length of the fiber, and so it is looking at the short association fibers that I've talked about. And the second is by looking at community organization. And by community organization, what I mean is these modular communities that develop through childhood and are important for attention. The way that you can think of them is that they are brain areas that are preferentially connected to each other and are thought to have specific functionality. So we hypothesize that short superficial fibers are particularly vulnerable to brain injury and that these fibers play an important role in intra-community connections. Therefore, we hope to evaluate the effect of traumatic brain injury, of mild traumatic brain injury, that is, on short-range fibers, and to probe the impact of brain injury on network structure with a focus on network segregation. So the way that we're going to do this is by utilizing the Adolescent Brain Cognitive Development Cohort. So this is not a clinical sample. This is a convenient sample of 10,000 youth from the United States that are going to be followed for 10 years. And so they're supposed to represent the distribution of the population. So after removing children who failed our pre-processing and matching on age, sex, puberty, family income, a variety of metrics, we were left with a population of 324 children with mild traumatic brain injury and 339 youth that were matched to this cohort. So the only thing that is different is the fact that this cohort has traumatic brain injury. The way that we're performing our analysis is by taking multi-shell diffusion weighted imaging and then performing particle filtering tractography. In the interest of time, I'm not going to go into too much detail on the methodology, but you can be sure to ask me after if you're interested. We then perform parcellation using the DKT atlas, which produces about 70 regions of interest, as you can see in this figure number 3. And then the last thing that we did is we created a connectome. So essentially we're creating a map of FA values that connect the various ROIs to each other. We are performing our statistical analysis by first doing a 3-way interaction for our group comparisons. So we're interested in the effect of group, obviously, on these metrics of interest, but also how sex and the age of the children moderates these relationships. If the 3-way interactions are not significant, they're trimmed from the model, and then we evaluate the 2-way interactions of group and sex and group and age. Similarly, if none of those are significant, they're trimmed from the model. So essentially, we seek to find the most complex model possible, and if no interaction terms are significant, then they're trimmed from the model. We do the same thing with our within-group analysis, but obviously without the group effect. So in our first study, we seek to establish the developmental trajectory as well as the effect of traumatic brain injury on short association fibers. So the way that we're doing this is by taking our connectomes that are weighed by FA. We're then thresholding them to only keep our short association fibers. So the way that we're doing that is by selecting fibers which are less than 85 millimeters in length and which only connect within the hemisphere. And this definition is based off of previous research that looks at short association fibers in other populations. So we then take our thresholded short association fiber FA matrices and perform our analysis using these connectomes. So the first thing that we did is we looked at the relationship between age and short association fiber development in only typically developing youth to understand how this fiber changes over time. And we find that, as you may expect, there is a positive relationship between time and mean FA. The next thing that we do is performed our group comparisons to understand the effect of traumatic brain injury on this fiber type. And what we found was that females with MTBI present higher superficial white matter FA than our males with traumatic brain injury. We also found these very strong, significant group differences such that our TBI group features elevated superficial white matter FA compared to uninjured males and females. We find this interesting relationship between age and our metric of interest when we looked at attention problems. So what we found was that our youngest concussed children displayed a reversed relationship between short association fiber FA and attention. So the red line is our youngest children, and we found that for higher values of FA, so that the way that we would expect it to move for typical development, that this is related conversely to more problems or more attention problems. So to summarize, our typically developing children display increases in superficial white matter FA with age. Children with MTBI displayed elevated superficial white matter FA, and females are particularly affected. And in young children with MTBI, elevations in our metric of interest consistent with normal development are associated with attention problems. The next thing that we did is we established the effect of traumatic brain injury on network segregation. So the way that we did this is we took our connectomes, this time weighted by streamline count, and I can describe why later if anyone is interested. And we use these to calculate metrics of community structure. So the metrics that we're using here, are modularity and mean clustering coefficient. Modularity basically describes the extent to which communities are connected within each other versus to other communities, so how cliquey a community is. And clustering coefficient basically describes how cool your friends are. So how connected are the nodes that one node is connected to, how well are they connected to others? And these are metrics of community structure. So what we found conversely is that our females present lower modularity. Our females with TBI present lower modularity than our males with TBI, and that our youth with MTBI present lower modularity. We found the same thing when we investigated our clustering coefficient, such that our TBI youth have lower values. Finally, when we looked at our relationship between our attention problems and our outcome metric, which is attention problems, we found that young concussed children also display this opposite from anticipated relationship with attention problems. So in summary, our children with MTBI display delayed network segregation, and females are particularly affected. And in young children with MTBI, reduced network segregation is associated with attention problems. So finally, we wanted to understand why this might be happening, and why our results for our short association fibers and our graph theory analysis don't seem to agree with each other. And so the way that we did this is by investigating the characteristics of our communities. So we wanted to understand what was the proportion of connections within a community that were short association fibers, and what was the proportion of connections between communities that were short association fibers. And remember, we hypothesized that within communities, short association fibers are very important. So what we found was that on the left side here, there's no difference between our groups in the proportion of within community connections that are short range. What we find is that our MTBI group has significantly elevated short range association fibers that connect between communities, which is not what we think these fibers should be doing. So to conclude, our MTBI youth displayed premature development of short range fibers and delayed development of network segregation. These structural aberrations in young children with MTBI were associated with attention problems, and a greater proportion of short-range fibers that connect between communities may underlie our findings of reduced community network structure. And I would like to thank my lab, labs that have helped me, C.E.T.S, and my committee, as well as the Brain Canada Foundation and the University of Toronto, and all of you for listening.
Sonja, thanks for presenting those extremely complicated analyses and results in such a comprehensible way.
Thank you.
And it sounds like you're really making some nice headway in terms of helping to us understand some of the results that we've got, some of the clinical results that we see out there.